let me tell you guys the way I use this. So the way I would use this is I look at whole communities. So I look at multiple species. You don't need to be writing this down. You just listen, and hopefully it'll make you understand a little bit. I look at whole communities of organisms. So I'll take a handful of soil, pull it up, and I want to know everybody that's in there. And there could be 1,000 species in there, easily. And so I want to amplify a particular gene. For me, it's 16S ribosomal DNA. So it's part of the ribosome of the bacteria. I amplify that particular gene, and then sequence it, and then I know that this fragment came from species A, this fragment came from species B. You can go into databases, GenBank, one of those things, and look that if I had the 16S sequence of this sequence, I know it's Bacillus subtilis, E. coli, whatever, something else that might be E. coli doesn't live in the soil, but you get my point. I lost your question. What was your question again?
It was basically how to choose primer.
How to choose primer, thank you. So it's pretty much, it's designed and known. So for me, I know I want a 16S primer. I know I want it to amplify that gene. So there's certainly times where you're coming through and you don't necessarily have a primer. And so then you'll be sequencing around it, and then you'll get to know that section, and then you'll design a primer for that. And that's more sophisticated molecular biologists would do that. Obviously, I would never do that, but there's other faculty over there that do that would be certainly looking for a gene in question. Not necessarily knowing what it is, and then they have to design that primer.
And is this based on one organism or multiple organisms?
In this case, it could be either. It really could be either. Good question. So she said, is this based on one organism or multiple organisms? And it could be either. So I absolutely can use this method for what I'm doing, and it's multiple organisms. And I could do this 1,000 different times just in one soup. I have the primer for the 16S region, and it's going to hit any organism I have in there that had the 16S gene. The organism's not in there anymore, it's DNA. Or if I want to amplify I want to amplify one individual organism's gene for, it's Lactoperum. You could do that. You could find the primer for that and amplify that gene for that organism. Yeah.
Why doesn't, when it's running this, why doesn't it, like, say for the C, why doesn't it stop at the first?
Stop it every time. Yeah. And that's something that I think has always mystified me, that it's not going to stop at every time. Sometimes it is going to extend out. You don't have every single gene in there as a DD.
Oh, just most of it?
Yeah,
So
some
there's
fraction
still a
of it.
chance that.
So there's still a chance that some of these genes, look, there it gave you a C that didn't stop, but that C did stop.
Yeah.
So it ends up being a little bit random.
Okay.
But yeah, no, I should have explained that. It's a good question, and everybody asks that every time. It's a good question. Right, anyone else? All right. So what it ends up, so the data from this, anymore we don't really run these PDS sequencing gels. We don't do that anymore. Anymore we use a sequencing machine so that your DNA fragments are running through a capillary, and they get seen by a laser beam that knows if it's a G, a C, or a T, or an A because you put a fluorescent probe on it. And then the output will look like a series of peaks. And you'll get 4 different colored peaks. And every time you see a different colored peak, you know that indicates a G, or an A, or a C, or a T. And it looks more like this. It's just easier, it's automated, it's faster, it's all the above. Okay, so some of the things that can come out of this is comparative genomics. So I alluded to this before, and I was kind of describing more what I do, but in a lot of different situations. In comparative genomics is where if you get the genome sequenced, or the 16S region, some ribosomal RNA region, DNA region of a cell, then you can compare that to other organisms in terms of how closely related they are. And you can understand their phylogeny. Who knows what the word phylogeny means? Okay, it's the relationship of the different organisms. Okay, their evolutionary relatedness. All right. So, hang on, I think I've even got it in here. Shoot, I don't. Okay. I can tell the evolutionary, it is in the next slide. That's right, it's in the next slide. The evolutionary, if I know the sequences of all of these different organisms, I can calculate how closely related they are to each other, how closely evolutionarily related they are to each other. All right, and you can do that for whole groups of organisms. All right, and we call that metagenomics. So meta meaning, we're looking at everybody looking across things. Looking at the genome of A, the genome of B, the genome of C, etc. And making big comparisons across that. can do this from anything from your own intestines to tube worms living down in the ocean, etc. And you can put all of the whole genomic extractions into one soup, cut that up into pieces of DNA, clone it into vectors, transform it into cells, and then sequence it and know what it's doing. And then go as far as if you're interested in a gene in question and a function in question, and you can even clone that into a cell and, stimulate it to express that gene, and then see the functionality there. All right, okay, questions on this before we move on to the next lecture? It actually extends really nicely from what I was just saying. Okay, so this actually segues really nicely into evolution. And the logical step from evolution with respect to microbial cells and macroorganisms as well is diversity. Why are there so many different types of cells? Why are there so many different types of beings in general? The bacteria in particular are really hyper diverse. Archaea as well, hyper diverse. And I've alluded to that throughout the semester, and now we're going to talk a little bit about why. You kind of understand why. You understand lateral gene transfer. But also how you measure that. We just got through that a little bit. We'll go into that a little bit more now. And then how that plays out. Just a really, in the next, finishing up today and then getting into Thursday, it's going to be a very coarse overview. Coarse in the sense of not highly resolved, not very fine-grained, very rough overview of microbial diversity. Okay, so phylogeny. Again, here it comes again. It's the evolutionary relationship of organisms. Anymore, it's based on a direct comparison of their genetic code. I can compare my genome to Yvonne's genome, and we can see how closely related we are to each other. Okay? I can take my same genome, I could pick a gene that's conserved across, say, all mammals, and I could see how closely related I am to a dog. Okay? I could pick a gene that's conserved across all vertebrates and see how closely related I am to a lizard. Okay? You can compare these genes and see how closely we match up. All right? In past times, it was much more based on things like physical appearance, physical traits, functional traits. Okay. And we rank these things. There's a hierarchy. Okay? The highest rank is domain. And the three domains that you know well in this class are the bacteria, the archaea, and the eukarya. Within domains, this should all look abundantly familiar, you guys. Within domains, you have phylum, class, order, family, genus, species. Okay? And a hierarchy. A lot of microbes have subspecies and strain levels. Okay, so let's just look at a classic case of this. In bacteria, one of the phylums is called the Proteobacteria. I'm going to go through those a lot in the next 2 lectures. And within the Proteobacteria, they're classified into alpha, beta, gamma, delta, and epsilon Proteobacteria. In this case, we're basically narrowing down from the domain bacteria all the way to a Shigella species. So, Shigella are within the Proteobacteria, and specifically, they're within the gamma Proteobacteria. Okay. Within the gamma Proteobacteria, you have all these different orders. Legionellas, you've heard of Legionella. Pseudomonads, you've heard of, you've been dealing with those in lab. Vibrios are interesting, I'll tell you a little bit about those in the next 2 days. Enterobacteriales, the Enterobacteria. Enterobacteria are the ones that live in your gut, the gastrointestinal organisms. Shigella is one of those. So in the family, Enterobacteriaceae, all right? In that, you have a lot of things you've heard of. Escherichia, Escherichia coli, please tell me you know your E. coli is Escherichia coli. Enterobacter, that's another one from your gut. Klebsiella, that's one from your gut that makes you sick. Proteus, you've been dealing with in the lab. Salmonella, you know what that makes you sick, etc. Yersinia, anyone know what Yersinia pestis is?
Black plague.
Plague. Shigella causes dysentery, if you're interested. So here we have Shigella at the genus level, and then down at the species, we have all kinds of Shigella species now. And then you can even have subspecies and strain numbers. So if, for instance, if I were to order E. coli from a catalog from ATCC, I'd have to pick from probably 1,000 different E. coli strains alone. Okay? Hierarchical, just like with macroorganisms. All right, so the definition of a species with respect to microbes, yes, we do it. I just spent an entire slide telling you about how we narrowed down to different species levels, and those species levels have names. And in the microbial world, certainly the lab-based microbial world, they all have strain numbers and strain levels. But the fact of the matter is the species definition in microbiology is really tricky. We don't like it. So based on the last 2 lectures I've given you, why don't we like the species definition in microbiology?
Quickly change. They can quickly change
Quickly change, absolutely. How?
via assimilating of their DNA.
Lateral gene transfer. They can quickly change through lateral gene transfer. So what I've said before is that sexual reproduction is the vertical transfer of genes onto your next generation. Bacteria, they don't sexually reproduce, but they do vertically transfer their genes onto their daughter cells. But what they also do is lateral gene transfer, horizontal gene transfer, such that they're transferring amongst, can be a completely different species, completely different phylum of organism. And they'll share their genes. They can just get taken up in the environment. So the definition of a species is really tricky. We use it, we need it in the lab, but it's a lot more loosely interpreted than it is with respect to macroorganisms. Okay? So if we were to go through the taxonomy of microorganisms, as I said, primarily we do it with respect to their DNA. Almost completely done by genetic code. But you can also look at things, and you guys are going to get a chance to do this at the end of your lab. You're going to look at some of their physiological and biochemical traits. In the end of your lab, you're going to take the streaks you've done, those unknowns that you've streaked from around the building, and you're going to go through and you're going to do a bunch of biochemical tests on them to try to identify who they are based on their biochemistry. All right? You can also look at their morphology. You're going to do a gram stain on them to see what they are, to see that they're gram negative or gram positive, and at least know that much. And then you're going to do these biochemical tests. There's other things you can do that you guys aren't really going to do in the lab, but think about how they interact ecologically in the environment that might help define who they are functionally and identify them. All right, and then obviously the genetic level, looking at their DNA sequence. So one thing you're going to do in the lab is you're going to take, I don't know if Dean has already told you this, you're going to take your streak plates that you've streaked all your unknowns in. Each one of you should have done that. You're going to pick one favorite one from your table. Pretty sure table level, not pair level. I think we're doing it in 4s. You're going to pick your favorite streak plate that's most isolated from the table. You're going to do a gram stain on it to identify if it's gram negative or gram positive. You're going to expose it to a whole series of biochemical tests to help identify how it handles things like, does it degrade lactose, does it degrade glucose, do all these different things. That's going to help you narrow it down. And then we're also going to PCR that little cell. We're going to PCR the 16S region of that. Ideally, hopefully get it back into a database and you can look at what the probable identification is. And won't it be incredibly cool if your genetic identification of this cell matches your biochemical definition? But that's dreaming. We'll help. Anyway, there's a lot of different ways to create the taxonomy and identify cells. All right, so going back to, that was kind of a segue, going back to the definition of the species being tricky and how we don't think of our own species definition in the same way that you can think of microbial definitions is one reason. It's not just, he's absolutely right, they share DNA freely, making what determines a species really, really tricky. And they're constantly changing because they can recombine their DNA so much, they have so much opportunity to recombine their DNA. And they can reproduce faster. But also, it's just the way we've defined them. So, I think our genetic code overlaps with that of a chimpanzee. It's something like 99%. So in terms of if I were to completely sequence a chimpanzee and completely sequence a human, our DNA would hybridize, at about 98%. We are almost genetically identical. Very little variation. If you were to do the same thing between a human being and a dog, it's something like 95%, 80%, 90%. It's really, really high. We are very, very genetically similar to any mammal. Okay? In bacteria, even within the same genus, we're not even in the same genus with the chimpanzees, certainly not with our dogs. But they'll have, even within the same genus, if you, Neisseria meningitidis, if you hybridize its DNA to itself, 100%, so you get the point, okay? But if I take Neisseria meningitidis and hybridize it with gonorrhea, yes, the same gonorrhea that causes gonorrhea, it would only hybridize at 78%. And then other species even less, 45 and 35%. So within the genus Neisseria, between meningitidis and flava, they only hybridize their DNA at 35%. Even within the same genus. So we're far, far more closely related to a chimpanzee than many bacterial species are to strains within their same species. All right, so the species concept is just, not only is it problematic with thinking about the way what defines bacterial species, but it's also, we're not looking at the same level of resolution. Okay? In macroorganisms, it's really highly resolved, partly from a human-centric standpoint. In microbial species, it's not. And we couldn't afford for it to be because they're too diverse. All right, so just going back to phylogeny one more time, and I'm going to tell you loosely how we calculate a phylogeny. So this is just an example, going back to dogs and macroorganisms. Phylogenetic tree is just the relationship of organisms to each other in an evolutionary context. All right, so here's a really simple phylogenetic tree where you have, gosh, what else do you have on this? You have mammals over here, birds and crocodiles are very closely related, snakes and lizards, turtles are kind of an offshoot, and then you have some amphibians here, frogs and salamanders. But notice that, and then some fish. Amphibians are much farther away from the mammals, let's say, than the birds are, etc. There's a distance away from these. And you can see this too for a bunch of different species of dogs, canids. All right, where to some degree you have the Arctic fox is very closely related to that little red fox, which everyone is. But then they're much more farther related than, say, to your boxer that's living in your backyard. All right, it's a map of the evolutionary relatedness of organisms. Okay, and as I said, you can do that from functional traits like biochemical tests. You can also do it from molecular hybridizing DNA. And this is the entire tree of life for all those domains. And let me just remind you that you have the three domains of bacteria, archaea, and bacteria. And we're all some distance related to each other. But to give you a feel for the resolution of this thing, which I said in the very beginning of this class, but I'll say it again now that you have a little bit more information, is that human beings, Homo, sit right between some sort of mushroom and corn, in the sense that if you were to resolve this thing, that's how coarsely resolved it is. And there's many, many different bacteria that are way farther apart from each other than we are even from the mushroom. No, okay, here we go. Yeah, that's all. Okay, so how do we calculate this? How do we calculate the distance between this eukaryote and this eukaryote? And we've got bacteria and that bacteria. All right? And you can do this using molecular data and something called a distance matrix. All right, so let me just tell you now, if you're studying, don't worry about this part. Okay, don't worry about that side of it. And we're going to step through how you calculate the distance between different species. So in this case, we have 4 different species of microbe, and we want to know the evolutionary distance between species 1, 2, 3, and 4. All right? If it was just 1 and 2, we can compare it directly. All right? But if we're trying to account for all 4 species, we need to do a matrix. All right? So what they've done here is we have a distance matrix between species 1 through species 4. And we know the sequence of some gene, and we know 15 nucleotides for this little cartoon example. In most cases, like for instance when I amplify the 16S, it's 1,400 nucleotides. All right, in this case, just for the purposes of this exercise, I have 15. So we know the sequence here of microbe 1 is this sequence. Microbe 2 is that one, 3, 4, etc. So we can go through and compare and go through the sequence of microbe 1 and the sequence of microbe 2 and pick out every time they're different. So at position 2, you have a C instead of a G there, so that's one difference. Same. At position 6, you have a G and a C. Again, that's different. That's two differences. At position 10, a U instead of a G. That's your third difference. And then at position 12, you have a C and a G. Again, that's different. So you had 4 differences out of 15. So if they were to hybridize microbe 1 and microbe 2 on that 15 base pair sequencing, they would find 4 out of 15 are different. At distance, they are that much apart. They are 4/15ths apart from each other. So you come down here in your matrix, and you put 0.27, okay? Or 4/15ths. If I were to ask you this on an exam, 4/15ths is fine. All right? And you can do the same thing with 2 and 3, and go through and find all the differences you see across there. And 2 to 3 is again 4/15ths, I believe. Yeah. 4/15ths, 0.27. So you do that for all of them, and then you go through and then you complete the matrix. All right? 1 and 1 are 100% similar. This is actually, in my opinion, slightly flawed. So if you do it on a test, I'll be flexible with that answer. But this is a distance matrix. The distance between 1 and 1 actually isn't 100% similar. It should be 0. In my opinion, I don't like this in the book. One of the few things I dislike in the book. But don't worry about it. If you study straight out of the book, when I've tested on that in the past, I accept both of them. But what you do need to know is be able to calculate these distances, these numbers. So if I compare 2 to 4, I know they're 1/3 apart from each other, etc. All right? Once you have those numbers, then you can go through and you can create a tree. So the good news for you and for your exam, this is something that's typically done by a bunch of nice software. You don't have to do that. Software does that for you. In the old days, they did it and they did the calculations. But to some degree, I could ask you to create a distance there where you put 3 and 4 are the most closely related.